This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas. Here we are again, trying to make sense of SARS-CoV-2. Seems like, just when you think things are settling down, a whole new batch of variants starts making noise.
It really doesn't stop, does it? The virus just keeps changing. And, well, our job is to follow those changes and figure out which ones we really need to pay attention to, and importantly, why.
Exactly. And that's what this deep dive is all about. We're going to look at some specific variants that have been causing a bit of chatter recently. Names like BA.3.2, NB.1.8.1, XFG. There are a few others too. We've been digging into the latest research, the papers trying to pin down their actual biological characteristics, their strengths and weaknesses, basically.
Right. And the aim isn't just to list off variant names. It's to really unpack the science, to understand, based on the biology, which of these variants might actually take off and spread, and which ones might just fade out. Maybe all bark and no bite, as you said.
Yeah. Think of this as your deep dive into the source material. We've sifted through it to pull out the key stuff, the things you need to know to get up to speed quickly on what the science is suggesting right now. Okay, let's jump in.
Okay, so the background here is what people are calling this variant soup situation. For a little while now, most of what we've seen circulating are descendants of BA.2.86.
Ah, right, Parola. That one caused a stir initially because it looks so different.
It did, and it's led to a whole family of variants. Now, the research we're focusing on uses LP.8.1.1 as its main reference point.
Why LP.8.1.1?
Well, it's another BA.2.86 relative, and it's been one of the more dominant strains circulating lately, so it makes a good benchmark.
Okay, so LP.8.1.1 is our current yardstick, and we're watching these newcomers try to, outcompete it.
Precisely. And the research does highlight several variants that are showing signs of having a growth advantage over LP.8.1.1. These include MB.1.8.1, LF.7.9, XEC.25.1, XFH, and XFG. They're popping up, increasing in frequency.
And then there's the one that got a lot of early buzz, BA.3.2, because it looked so dramatically different.
It really did. BA.3.2 is interesting because it's what they call a saltation variant.
Saltation?
Yeah, it means it took a really big leap, mutation-wise, like over 50 mutations compared to its ancestor, BA.3, and about 44 differences compared to our reference, LP.8.1.1. So when something changes that much and pops up somewhere new, like it did in the Netherlands back in April 2025 after first being seen outside South Africa, well, it gets noticed.
Wow, over 50 mutations. That sounds like it should be, really concerning, right? Like a major upgrade for the virus.
It sounds like it should be, yeah. On paper, it looks alarming, but this is exactly where the research helps us dig deeper, because that high mutation count, it doesn't automatically mean it's a successful variant.
Okay, so let's get into that. What are the key things the scientists actually measure to figure out if all those changes truly make a variant better at spreading?
So the research really boils it down to two critical factors. Yeah. First, how good is the virus at actually getting into our cells? Our cells have that ACE2 receptor like a doorknob.
Right.
The virus uses its spike protein like a key to grab onto that doorknob and get inside. So factor one is how efficiently does its spike protein bind to ACE2? How well does the key work in the lock? Let's call that infectivity or cell entry efficiency.
Okay, step one, can it actually get in the door effectively?
Exactly. Then step two is immune evasion. How good is it at avoiding the antibodies we already have, from past infections or vaccines? Can it sneak past our immune system's defenses?
Get in the door and dodge the guards.
Got it. So how did the researchers test these new variants on those 2 points?
They use a range of lab techniques. Things like pseudoviruses, basically harmless viruses engineered to have the spike protein of the variant they're studying to see how well they enter lab-grown cells.
They directly measure how strongly the spike protein binds to the ACE2 receptor protein. And really importantly, they test neutralization. They take blood serum or plasma from people or mice with known vaccination or infection histories and see how well the antibodies in that serum can actually block the variant virus in a lab dish. They also test specific lab-made antibodies, monoclonal antibodies, against them.
Okay, quite a thorough workup. So let's go back to BA.3.2, the flashy one with all the mutations. You called it a paradox.
It really is, because despite all those mutations and being quite good at dodging antibodies, the research found its ability to actually infect cells, its infectivity, was surprisingly poor in their tests.
Really? Weak infectivity, how does that work if it's got so many changes?
Yeah, it's fascinating. So they looked closely at the spike protein, the very tip of the spike, the part called the RBD that actually makes contact with ACE2. But okay, similar strength to LP.8.1.1 actually. But when they looked at the whole spike protein and how efficiently it engages with ACE2 on a cell surface, BA.3.2 was the worst performer, the lowest ACE2 engagement efficiency among all the variants they tested.
Huh. So the key tip fits the lock okay, but maybe the rest of the key is shaped funny, or it's hard to turn, making it difficult to actually open the door.
That's a really good analogy. The researchers think it might be because the BA.3.2 spike tends to stay in a more closed shape, with that binding part tucked away more often. This makes it less effective at grabbing onto the ACE2 receptor when it encounters a cell.
And the result of that difficulty getting in?
Well, the paper uses terms like disastrously low fitness or infectivity compared to LP.8.1.1 in their lab assays. Basically, if you struggle to get into cells, you can't replicate efficiently. And if you can't replicate efficiently, you're probably not going to spread very well in the population, no matter how good you are at hiding from antibodies.
But you said it is good at hiding. That's the other part of the paradox.
Absolutely. The data confirms BA.3.2 is a strong immune escape artist. Antibodies from vaccinated mice didn't neutralize it very well. And in tests with human plasma, well, it showed really significant evasion.
How significant?
The study found, for instance, it took about 3 to 4 times higher concentration of antibodies in the tested plasma to neutralize BA.3.2 compared to neutralizing LP.8.1.1, and compared to its older relative, BA.3, it was something like 11 times harder to stop.
Wow. Okay, so antibodies really struggle with it. And you mentioned specific types of antibodies, those monoclonals.
Yeah, it was remarkably resistant to a group called class 14 monoclonal antibodies.
Yeah.
These are usually quite potent, quite broad against many Omicron variants. BA.3.2 has a specific mutation, K478N, that helps it escape antibodies binding near that spot. There was also an interesting side note about how class 14 antibodies might be more common in people who had inactivated vaccines versus mRNA ones, which could mean different immune backgrounds might handle BA.3.2 slightly differently.
Okay, so summing up BA.3.2, amazing at dodging immunity, but apparently pretty bad at the crucial first step of getting into cells. What's the verdict then? Is it going to be the next big wave?
Based on this research, probably not. That low infectivity is a major bottleneck. Its potential to become dominant seems quite limited, at least in its current form.
Unless it changes again, fixes that infectivity problem.
Exactly. That's the caveat. If it were to pick up additional mutations that somehow stabilize its spike in a more open or receptive conformation, improving its ACE2 engagement, well, then the game could change. But as it stands, its high evasion alone doesn't seem to be enough.
Right. So BA.3.2, lots of bark, maybe not much bite for now because it trips at the first hurdle. Okay, what about the others? The ones you said are actually showing growth advantages, like MB.1.8.1, XFG, LF.7.9.
Yes, let's turn to them. These are the variants the data suggests are genuinely increasing their share compared to LP.8.1.1 out there in the real world.
And they seem to have figured out the formula better.
It looks that way. And interestingly, the research points out some convergent evolution going on. Meaning different variants are independently hitting on some of the same beneficial mutations. Changes like Q4N3E, A475V keep popping up on the spike protein in these different growing lineages. It suggests these specific changes are giving them an edge right now.
Okay, let's spotlight MB.1.8.1. I think I saw it nicknamed Nimbus.
That's the one, yeah. Coined by P. Ryan Gregory.
Yeah.
And MB.1 by K.1 is really interesting lineage-wise. It's not just another branch off the BA.2.86 parole tree like LP.8.1.1.
Oh.
The research identifies it as a triple recombinant. It's actually stitched together from genetic pieces of both the BA.2.86 lineage and older XBB relatives like XBB.1.9 or XBB.1.19.
So it's a hybrid of different major past lineages, and it's actually spreading.
Yes, the paper notes it gained traction significantly in parts of Asia and is now found globally, enough that the WHO designated it a variant under monitoring back on May 23rd, 2025.
Okay, so it's demonstrating real-world success. What about its biology, according to this research? Does it have DA.3.2's problem with getting into cells?
No, and that seems to be key. Unlike BA.3.2, the research found NB.1.8.1 maintained high ACE2 binding affinity and showed robust ACE2 engagement. It gets into cells efficiently in their tests. Its infectivity looks pretty good.
Okay, so the getting in the door part is sorted. What about dodging the guards? Immune evasion.
It's got that too. It shows significant humoral immune evasion. Antibodies in the tested human plasma found it about 1.5 to 1.6 times harder to neutralize compared to LP.8.1.1.
So noticeably more slippery than the current baseline.
Right, and it has specific mutations contributing to that. A change at K478I helps it evade certain antibodies, and another one, A435S, seems to reduce neutralization across several antibody targets.
So NB.1.8.1 seems to have both parts of the equation figured out, good infectivity and good immune evasion. What does the research conclude about its potential then?
The conclusion is pretty clear. This combination makes it a strong contender. Its balanced profile of retaining good cell entry while also having enhanced immune escape gives it significant potential for future prevalence. It's definitely one to watch closely.
Okay, Nimbus seems like a well-rounded competitor. What about the other growers, like XFG and LF.7.9? Are they more like Nimbus or more like BA.3.2?
They're kind of in between, or maybe closer to BA.3.2 in one respect. The research suggests that while they do show strong immune evasion, they might also be facing some challenges with ACE2 binding and cell entry. Not necessarily as bad as BA.3.2, but still a potential hurdle.
Okay, tell me about XFT first.
So XFG is another recombinant. It has specific spike mutations, H445R, N487D, Q493E, T572I, that make it quite evasive, nearly twice as hard to neutralize as LP.8.1.1 in the tests, and resistant to certain antibody classes, partly due to N487D and Q493E.
Sounds very evasive.
But that N487D mutation, while helping with escape, seems to reduce how well the spike binds to ACE2. So XFG showed markedly lower ACE2 binding affinity and lower overall ACE2 engagement compared to NB.1.8.1 or LP.8.1.1.
Ah, so another case of a trade-off. Great escape, but maybe pays a price in entry efficiency.
Seems like it. The research suggests it might need compensatory mutations, other changes to improve that ACE2 interaction if it's going to sustain transmission effectively long-term.
And LF.7.9, similar story.
Pretty similar, yeah. It has mutations like A475V and H445P. It also shows strong immune evasion again, about 1.5 to 1.6 times harder to neutralize than LP.8.1.1. And that A475V helps it resist class 1 antibodies.
But the downside?
The A475V mutation also seems to reduce ACE2 binding affinity, so like XFG, LF.7.9 showed reduced ACE2 binding and lower cell entry efficiency in the tests, again suggesting it might need further adaptation to optimize cell entry.
So these fast growers, XFG and LF.7.9, might be getting an initial boost from their strong immune evasion, but they could hit a ceiling if they don't improve their ability to get into cells.
That's the implication from this research, yes. It highlights just how complex this evolutionary landscape is. The paper also briefly touches on others like XAC.25 on point 1, which apparently does have robust AC2 engagement, and XFH, which has low engagement, just showing the diversity strategies being tried out.
This is really fascinating, breaking down the specific mechanics, but let's connect it back directly to you, our listener. Why does understanding this level of detail matter?
Well, I think this kind of deep dive helps us look beyond just the headline saying new variant detected. It gives us the tools to understand the why some variants, even those that look scary initially because of mutations like BA.3.2, might not actually become dominant, and why others, maybe less dramatic-looking ones, are the ones actually gaining ground.
It's about understanding the underlying biology, not just the number of mutations.
Exactly. This research really underscores that a successful variant needs to win on two fronts. It has to be good at evading our immunity, and it has to be good at physically getting into our cells. You need both. One without the other seems to really limit a variant's potential.
Did the research mention anything about how current or upcoming vaccines might hold up against these?
It didn't directly test vaccine effectiveness in people. That wasn't its focus. But it does provide data on how antigenically different these variants are from the lineages the vaccines target, like JN.1 or KP.2. And while these new variants, particularly NB.1.8.1 and BA.3.2, are quite distinct from JN.1 and XBB ancestors, there's still likely to be some cross-protection, especially against severe disease, from current vaccines. The key mutations they have, some are becoming quite common across different lineages. The research mainly focused on measuring the relative evasion.
Yeah.
How much harder are these new variants to neutralize compared to the currently circulating LP.8.1.1?
So the vaccines probably still offer a base level of protection, but these variants are definitely shifting the goalposts for antibody neutralization. And understanding this balance infectivity versus evasion, that's crucial for public health.
Absolutely. Knowing which emerging variants have that winning combination, that balance of efficient entry and strong evasion, like ND1.8.81 seems to have, that's critical intelligence. It helps inform surveillance, risk assessments, and basically keeps us better prepared for what the virus might do next.
Okay, so let's recap this deep dive. We looked at BA.3.2, ton of mutations, great immune escape, really looked like a contender. But the research shows it seems to be tripped up by poor infectivity, difficulty getting into cells. Might be all bark, no bite for now.
Right. Its Achilles heel seems to be ACE2 engagement.
Then we looked at the ones actually growing, like NB.1.8.1, Nimbus. It seems to have found a much better balance. It's still good at getting into cells, and it has significant immune evasion. That makes it a variant with real potential.
A more complete package, biologically speaking.
And others like XFG and LF.7.9 show strong evasion too, but might share some of BA.3.2's challenges with efficient cell entry, possibly needing more evolution to really compete.
Yeah, the data clearly shows immune escape is necessary.
Yeah.
It's definitely not sufficient on its own. That efficient cell entry is equally vital for a variant to really take over.
So here's something to mull over, building directly on that idea. The research strongly suggests that truly successful variants need both high immune escape and high infectivity. It's a tough balancing act for the virus. Given that, how might a variant like BA.3.2 evolve to overcome its current weakness in getting into cells? Could it find that fix? Or is it more likely that its moment has passed and the advantage shifts to variants like NB.1.8.1 that already seem to have struck that balance? What does this constant evolutionary trade-off tell us about where SARS-CoV-2 might head next? Something to keep thinking about.
Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other podcasts, responding to the content, and checking out our related articles at helioxpodcast.substack.com.